This is me Arzu here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 2 video of the oral mucosa or the oral mucous membrane, and in the previous video of this oral mucosa we have just discussed about the introduction to this oral mucosa. Okay, we have discussed with the what is the oral mucosa. Okay, so we have just discussed with the introductory part to the oral mucosa, and in this part 2 video of this oral mucosa we will be discussing with the functions of the oral mucosa and about the keratinized oral epithelium. So we will, in this keratinized oral epithelium, we will be discussing with the different layers. Okay, so we know that we have 4 different layers in our epithelium. Okay, in keratinized oral epithelium: stratum basal, stratum corneum, okay, stratum granulosum. So we will be discussing with all those layers in this video itself, and in upcoming videos we will be discussing with the non-keratinized epithelium and other remaining topics of this oral mucosa. And you guys know that this oral mucosa chapter is one of the biggest chapter of dental histology, so I am trying to include all those important points in my notes and also trying to explain all those concepts to you. Okay, so video may be long, but I will try my best to explain all those points to you. Okay, and also in the previous videos of the dental histology, okay, so we have completed one chapter in 3 parts, but in this chapter I think it will take more than 3 parts because this chapter is a bit larger. Okay, so without delaying much, now let's get into our topic. So our first topic is functions of the oral mucosa. Okay, so our first function is the defense and the protection. Okay, so in defense and the protection, okay, so the mucosa is just the lining of the oral cavity. Okay, so simply the mucosa is the outermost lining, okay, lining of the oral cavity. So it must be functioning in the defense or in the protection. Okay, so how it functions and how it be a barrier. Okay, so that is discussed here. So in the first one, integrity of the oral epithelium. Okay, so it forms the effective barrier. Okay, so there is it forms a system. Okay, it forms a proper system that forms a proper effective barrier. Okay, after that, in the next point, oral mucosa is impermeable to the bacterial toxins, and after that also secretes the antibodies which has effective cell mediated and the humoral immunity. Okay, so this is important. So in this pattern, okay, oral mucosa is acting like a defense. Okay, it is defending the toxins. Okay, so in this pattern it just defends the toxin. After that, in the next point, it protects the deeper tissues from the compressing, stretching, searing and hard particles in the food. Okay, so it protects the deeper tissue from compressing force, stretching force, searing force and also the hard particles that are present in the food. Okay, so in this defense and protection function, you just have to remember that our oral mucosa, it forms a proper effective barrier, and it also secretes different kinds of antibodies that are effective in cell mediated, that are effective by cell mediated or the humoral immunity. Okay, so humoral means antibodies secreting type of immunity. Okay, so just remember this secretes the antibodies, and those antibodies try to keep us protected. Okay, it protects us, it defends the toxins. After that, in the next point, it protects the deeper tissue by different mechanical forces and also the hard particles in the food. Okay, so in such a pattern, it just defends and protect the deeper tissue of the oral mucosa of the oral cavity. And in the another function, there is a sensation function. Okay, so oral mucosa is sensitive to the touch, pressure, pain and temperature, better appreciated in the anterior part of the tongue or the mouth. Okay, so sensation means oral mucosa should sense the stimuli. Okay, so this sensation is also one of the function of this oral mucosa, and it's just sensitive to the touch, pressure, pain and the temperature. Okay, so they are the general sensation. And taste, okay, so taste is a special sensation, and it's also mainly felt in the anterior 2/3 of the dorsum of the tongue. Okay, so tongue is responsible for this taste sensation. After that, in another point, receptor for the reflexes like swallowing, gagging and salivation are also present in the oral mucosa. Okay, so those 3 points are important here. So you just have to remember it is sensitive to the general sensation and also to the special sensation. Also it have the receptor that are responsible for the swallowing, salivation and the gagging. Okay, after that, in the next function, that is a lubrication function. Okay, so in lubrication function, salivary gland secretion keeps oral cavity moist and prevents the mucosa from drying and cracking. Okay, so this lubrication function is also one of the important function of this oral mucosa. Okay, as it help in different functioning of the oral cavity. Okay, so during speech, also during chewing the food. Okay, so this lubrication is also very important. After that, in the last function, so there is thermal regulation. Okay, so this is not much important function of the oral mucosa, but in humans it plays a little role, and in the animals oral mucosa aids in the thermal regulation. Okay, so in oral mucosa, this in animals oral mucosa have also the thermal regulation function. Okay, so in this function section of the oral mucosa, okay, so I have discussed this in fast. Okay, and it is also important and it is also easy to understand, so I think you guys have captured all those points. So just remember, there is a first function, that is defense and protection function, another is sensation function, lubrication and thermal regulation. And thermal regulation is not much prevalent in the humans, but it is more prevalent in the animals. Okay, so we have completed the function portion of the oral mucosa. Now we will discuss with the keratinized oral epithelium. Okay, so before discussing that keratinized oral epithelium, so I have taken this proper table. Okay, and this table is very important, and you guys should remember this table. Okay, so this table will give you overview of the oral mucosa, okay, and the layers of the oral mucosa. And this table I have taken from the Orbans. So oral mucosa, okay, so we can divide it in the three groups. Okay, so our outermost part of this oral mucosa is the epithelium or the oral epithelium. Okay, so I have written some points about this oral epithelium. Okay, so oral epithelium, it is derived from the oral ectoderm. Okay, so this is important to remember. After that, it is stratified squamous epithelium, present either keratinized or non-keratinized. Okay, so epithelium that is present in this oral mucosa or the oral epithelium, it is a stratified squamous epithelium. So stratified, I think you guys know, you have been familiar with this term stratified. Okay, so there are different layers of epithelium, so that is stratified and squamous means there is a flattened polygonal, okay, squamous type of cells at the topmost layer of the epithelium, so it is a squamous and stratified epithelium, and it may be keratinized or the non-keratinized type. Okay, after that, in the next one, both keratinized and the non-keratinized consist two groups of the cell, that is keratinocyte and the non-keratinocytes. Okay, so I think many you okay have confusion in this. Okay, so you just think that in keratinized there is keratinocyte and in the non-keratinized there is non-keratinocyte. Okay, so that is properly wrong. So from today you should understand that either it is a keratinized oral epithelium or the non-keratinized oral epithelium. It consists the two groups of the cell. Okay, so there is keratinocyte and non-keratinocytes. Okay, so they both are present in both of this epithelium. Okay, so in the non-keratinized, now we have three layers. Okay, and in the keratinized we have four different layers. Okay, so in the non-keratinized we have stratum basal, stratum intermedium and stratum superficiale. After that, in keratinized we have stratum basal, an additional layer, that is stratum spinosum, stratum granulosum in the replacement of this stratum intermedium, and at the top we have stratum superficiale. Okay, so stratum basal and superficiale are common in both, but this stratum intermedium, okay, is different in this keratinized epithelium. Okay, so you can remember this by your own mnemonic, but I have prepared a simple mnemonic. So in keratinized epithelium you can remember by this. Okay, you can remember this as a bis. Okay, so bis or sib. Okay, anything you can remember like that. Okay, so sib means there is a god. Okay, Siba, so you can remember like that. Okay, sib, bis or sib. Okay, so you can sib So this V is B. Okay, I think you will be confused in this mnemonic, so you can prepare your own mnemonic, but I used to remember this by BIS. Okay, so BIS, basal, intermediate and superficially. And from this BIS, okay, this I intermedium, okay, so it is not present in keratinase, so I take this so I am bit clear in that. Okay, so basal and superficiale are also present in keratinase, and basal in the lower portion and superficiale in the upper portion. Okay, and additional two are spinosum and granulosum, but also you can prepare here a mnemonic, separate mnemonic, that is BSGS. Okay, so taking a first letter. Okay, so this is very easy to remember also, and I don't think you should have to be bothered in this remembrance. Okay, in remembering those layers, this is very easy to remember, I think. So you can remember just BSGS. Here you can remember by a BIS. Okay, so BIS means basal, intermedium and the superficiale, and here basal, and again spinosum and granulosum and the superficiale. Okay, so you can remember like that way. So BSGS, here BIS. So that is the mnemonic, and also you can prepare your own mnemonic. After that, in the next group, there is a basement membrane. Okay, after epithelium, so epithelium have basement membrane. So if this is a epithelium, okay, so if this is a epithelium, so this is a basement membrane. So this basement membrane is also called basal lamina ultrastructurally, and it have two parts. Okay, so one is a lamina lucida and another is a lamina densa, and I will be discussing this basement membrane properly in upcoming videos. So just remember now, there is one lamina lucida and there is a lamina densa, and that are the parts, that are the layers of the basal lamina basement membrane. After that, in the next group, below that basement membrane there is a connective tissue, and the upper layer we have lamina propria, below that there is a submucosa. Okay, so from this table we have came to conclusion that, okay, so there is a epithelium, then there is a basement membrane, and there is a connective tissue. So in epithelium also, if it is a keratinase, the first layer, so outermost layer is a superficiale. Okay, and this superficiale layer is also called corneum. Okay, stratum corneum. Okay, so I will be discussing this in later in this video itself. And superficially means most superficial layer. Okay, or stratum corneum. After that, after corneum we have a granulosum. Okay, so in granulosum there is a granules. Okay, after granulosum there is a stratum spinosum, and spiny formation, spiny processes are present there in this layer. After that is stratum basal. Okay, basal means at the base of the epithelium, and below the base there is a basement membrane. In basement membrane also there is a lamina lucida, then after there is a lamina densa, and below that lamina densa we have connective tissue layer, and in the connective tissue we have lamina propria, and below lamina propria we have the submucosa. Okay, so different kinds of blood vessel, different kinds of the salivary glands are present in this submucosa. Okay, but they are absent in this lamina propria, and we will be discussing with those in upcoming videos. So first of all, I think I will show you one picture, then it will be more clear. Okay, okay, so let me show you one picture of this. Okay, okay, so this is more clear. Okay, so this is a posterolateral lesion of the palate. Okay, so here you can see. Okay, so this much. Okay, so this much, so there are the ridges. Okay, so they are the ridges, the reti ridges we call them. Okay, so longer regular reti ridges. Okay, so those finger like projection of the epithelium are called reti ridges. Okay, so they are not plain. Okay, as like I have drawn here. Okay, so basement membrane and this is epithelium, so they are not just plain. And in the different parts of the oral cavity we have a different structure. Okay, so we have different types of junction between the epithelium and the lamina propria. Okay, some where there is a long reti ridges, some there is a smaller reti ridges present there. Okay, but they are in such a curvy pattern itself, they are not properly flattened. Okay, so here we can see the epithelium. So this is a epithelium layer, and between this epithelium's finger like structure there is a connective tissue. Okay, so this connective tissue is the lamina propria, and this is also the lamina propria. Okay, and lamina propria also we have two different parts. Okay, and that we will be discussing. So this is one part and this is another part. Okay, so there is a reticular part and there is a papillary part. Okay, so we will be discussing with that in upcoming videos. Lamina propria. After the lamina propria we can see the blood vessels. Okay, so vertical bands of the connective tissue containing the mucosa to the periosteum. So this is a periosteum, and below the submucosa we have periosteum. Okay, so periosteum is the outermost layer of the bone. So in this submucosa it contains the minor salivary glands. Okay, and in between them there is a connective tissue strands are present there connecting to the periosteum, palatal bone with the periosteum. Okay, so you just have to make a proper concept. Okay, so first of all there is a epithelium, after that there is a lamina propria. Okay, and after lamina propria we have submucosa. Also here if I am okay, so this picture is quite clear. Okay, okay, this picture is quite clear. So this is a keratinized epithelium. So in keratinized epithelium we can just see the first layer. Okay, so it is quite of a rough type of layer. Okay, so proper demarcation of cell you can't see here. Okay, because there is a presence of the keratin. Okay, so keratinization is present here in the uppermost layer, so they are called keratinized epithelium, and the outermost and uppermost layer is a stratum corneum, and they are parakeratinized. Okay, so about parakeratinization and the orthokeratinization I will be discussing that, but for now just remember outermost layer. Okay, so that is a stratum corneum here, and below that we can see the stratum granulosum. Okay, so this is a stratum granulosum layer. Okay, so it will be more clear here. So this is a stratum granulosum. Okay, below this stratum corneum, and below this stratum granulosum we can see the proper cells. Okay, so they are having the spiny processes and they are called stratum spinosum, and below the stratum spinosum we can see this single layer type of cells. Okay, so they are stratum basal. Okay, so they are the stratum basal, and below that this red line, okay, that is demarcating the epithelium and the connective tissue. This is a basement membrane. Okay, so now I think you are clear in this. Okay, so now I will move ahead. So I have written about all four layers of this keratinized epithelium in this note. Okay, so I will be discussing that. So in keratinized oral epithelium, so our first and the basal layer is the stratum basal. Okay, so the layer of the cuboidal or the columnar cell is present in this stratum basal. Okay, so cuboidal or columnar type of cells are present in this stratum basal. Also you can see in this picture. Okay, so they are quite of cuboidal or the columnar type of the cells. Okay, okay, so this picture is taken. So this book is a manual of histology. Okay, by the Mazijos. Okay, so you can refer to this book for looking those picture. Okay, but for now you just think this is a cuboidal or the columnar type of the cells. Okay, so in the stratum basal. After that it is a less differentiated oral cells. Okay, so it is not much differentiated into the any type into the any other type of the cells, so it is least differentiated. After that it lie on the basement membrane. Okay, so it is the basal and lie on the basement membrane, and it synthesizes the DNA and undergo the mitosis. And basal cells and the parabasal spinous cells are referred to as the stratum germinativum, but only basal cell can divide. Okay, okay, so this point is very important, you can just remember. Okay, so this basal cell and the cell that is above this basal cell, so that is a parabasal cell. They are particularly parabasal spinous cell. Okay, so the cell that is above this basal cell we have spinous stratum spinosum. So that parabasal spinous cell both together are called as the stratum germinativum. Okay, so both together they are called stratum germinativum, but only basal cell are responsible for multiplication and the division. After that abundant ribosomes and elements of RAR is present here. Okay, and as they are synthesizing DNA, undergoing mitosis, secreting proteins, so there must be ribosome and the rough endoplasmic reticulum abundant. After that in the next point there is a two populations of cell, and this is very important. One is a serrated cell and another is a non-serrated cells. Okay, so now we will discuss with this the serrated cell and the non-serrated cells. Okay, so first we will see about the serrated cell. So serrated cells, they are heavily packed with the tonofilaments. Okay, and they are for the adaptation for the attachment. Okay, so the main function of this cell, serrated cell, is the adaptation for the attachment. So that is the attachment. Okay, and I have discussed more about the serrated cell, but I'll first discuss with this. Okay, so non-serrated cell, they are slowly cycling stem cells and or the progenitor cells. Okay, and there are further classification in this non-serrated type of the cell, and they are stem cell and the amplifying cell. Okay, and in the stem cell, they preserve the genetic information to retain the proliferative potential. Okay, so stem cell, they just preserve the genetic information. Okay, they preserve the genetic information to retain the proliferative potential, and they are small in number and retains the proliferative potential. So same thing is written here and here. And after that, in amplifying cell, it provides the more number of maturative cell, larger compartment, larger in number, and increase in the cell number available for the maturation. Okay, so just you should remember that. Okay, so in this uh, stratum based cell, okay, so we have two different kinds of the cells. One is a serrated cell and another is a non-serrated cell. Okay, so and the serrated cell is mainly present in the stratum based cell for the attachment. Okay, so you need the attachment with the basement membrane, okay, for stabilizing the epithelium. So serrated cells are particularly present for the attachment, and so as they are present for the attachment, it should have tonofilaments. Okay, so the filaments should be present in this serrated cell in larger quantity, and in the non-serrated cell, they are not for the attachment, they are just a stem cell. Okay, so they just proliferate or they just preserve the genetic material, they just present the information, genetic information. Okay, so in non-serrated cell also there is a stem cell and another is amplifying cell. Okay, so one is amplifying cell, they just get multiplied. Okay, and after multiplication they just provide all those cell to the upper layer. Okay, so they increase in a cell number for the maturation, so they just increases the number of the cell for the maturation. And this stem cell, they are the smaller in number and they just preserve the genetic information. Okay, so they are responsible for preserving the genetic information and retaining the proliferative potential. But this amplifying cell, they do not preserve the genetic information and their main function is just to multiply. Okay, and provide the cell to the upper layer. Okay, so that is the difference between the stem cell and the amplifying cell. Okay, so the name also suggests the same function and you can easily remember that. And now we will discuss in more detail about the serrated cells. Okay, so serrated cells, they have protoplasmic processes called pedicles projecting from the basal surface towards the connective tissue. Okay, so connective tissue is just below the basement membrane. Okay, so those serrated basal cell have different processes as I have told earlier, and those processes are also called pedicles. They are projecting from the basal surface towards the connective tissue. Okay, piercing the basement membrane. After that, a round or oval nucleus which occupies the 1/3 of the cell volume, and nuclear membrane is smooth with the deep invagination. Okay, so this is just a morphology of the nucleus that is present in this serrated basal cell. Okay, and this is also morphology of this non-serrated type of the cells. Okay, but mostly this is a serrated basal cell and you can remember this. Okay, so round and oval nucleus is present there, occupies 1/3 of the volume of the cell, and smooth and with a deep invagination are present in the nuclear membrane. After that, in the next point, there we can see about the hemidesmosomes. Okay, and we have another desmosome. So hemidesmosome is the connecting complex. I think you have discussed in a general physiology about this. So hemidesmosome is the protein complex. Okay, that is just a protein and a filaments complex. Okay, that is attaching the cell with the basement membrane. And the desmosome is also similar with that of the hemidesmosome, but it is present laterally on the cells. Okay, so if this is a cell, okay, and if this is another cell and this is a basement membrane, so between the cell and the basement membrane there is a hemidesmosome, but between the two cells, okay, so this is a desmosome. Okay, so that is a desmosome. And about this desmosome and hemidesmosome, you can be properly studying. Okay, if you want to study about this in detail, you can just refer to the general physiology. Okay, so there is properly written about them, but for now in oral histology, so this hemidesmosome about on the basal lamina are found on the basal surface. So hemidesmosome about on the basal lamina found on the basal surface, and they contain the single attachment plaque adjacent plasma membrane and associated extracellular structure. Okay, so the content of these hemodesmosomes are single attachment plaque, okay, the plaque or protein, okay, and adjacent plasma membrane. Okay, so adjacent plasma membrane and associated extracellular substances present there, and laterally cells are connected by the desmosome, which contains the cell membrane, pairs of the denser regions, attachment plaques, as well as intervening extracellular structure. Okay, so there is a composition written here. Okay, so laterally cells are connected by the desmosome, contains the cell membrane. Okay, so cell membrane we have, and pair of the denser region, attachment plaque. Okay, so attachment proteins are present in either side of the cell, as well as intervening extracellular substances are also present there, with the tonofilaments attached to the attachment plaque. Okay, so simply you just remember, I am giving you a technique, okay, to remember this. So just think, this is a one cell, another, this is another cell. Okay, so between the cells, okay, there is a transmembrane protein present here. Okay, so one is transmembrane protein, another is a attachment plaque. Okay, so attachment plaque means they are attaching proteins. Okay, so they are present here. Okay, so they are the attachment plaques. Okay, and the attachment plaques, they connect the transmembrane protein with the filaments. Okay, so I will draw a filaments too. Okay, so this is a filament. Okay, so this is a filament present in the cells. Okay, so just to remember, we have three structure, this is second and this is a third. Okay, so first is a transmembrane protein between the cells, another is the attachment plaque. Okay, attachment protein, which is in blue color. After that there is a filaments present in the cell, that is in a red color here. Okay, so this attachment protein connects the filament with the transmembrane protein, and between this transmembrane protein, okay, there is a extracellular structures present there. Okay, extracellular structure means the structure that is present uh, in between the cells. Okay, so they are extracellular structure and they are present in this region. So just remember, transmembrane protein, attachment plaque, and the filament with the extracellular structure intervening in the transmembrane protein. Okay, so that is a proper structure, you can remember like that way, but I have written this from the Orbans. Okay, and you can refer this properly. If you want to read in detail, you can go to the general physiology and read about this. Okay, so there is a similar kind of physiology in oral mucosa too. Okay, after that, tonofilament, this I have discussed, and two principal type of the protein. Okay, so the transmembrane protein and protein inside the cell related to the attachment plaque or the cytoplasmic adapter protein. Okay, so the same thing I have written here. So this is a transmembrane protein, and this attachment plaque or the cytoplasmic adapter protein. Okay, cytoplasmic adapter protein is present here, and we have filaments. After that, this point is important here, gap junction are occasionally seen, tight junction are not observed in the oral mucosa. Okay, so gap junctions are mostly for the electrical communication. Okay, so these gap junctions are mostly present between the two cells for the communications, okay, and transfer of the material, but in oral mucosa it is not much required. So the gap junctions are also occasionally seen in oral mucosa. And these tight junctions, okay, so these tight junctions are for the proper and strong attachment between the cells, and it is not required must in this oral mucosa, and they are not observed. Okay, so they are not seen in the oral mucosa, and this is MCQ question that can be framed into the MCQ question. Okay, so we have completed about the stratum basal, and another thing, okay, so is the stratum spinosum. But before that, I think I have written a thing. Okay, so you should look over this also. Okay, before I will discuss with the stratum spinosum. So this is a turnover time. So this turnover time is time taken by the cell to divide and pass through the entire epithelium. Okay, so that is the turnover time. Okay, so turnover time means the time, it is a time taken by the cell to divide at the stratum basal layer and pass through the entire epithelium and reach the stratum corneum layer. Okay, and we have the turnover time different on the different places of the oral mucosa. And in the gingiva we can see it is 41 to 57 days. In the cheeks we have 25 days. In the skin 52 to 75 days, and in the gut there is 4 to 14 days. Okay, so it is smaller in the gut. Okay, so you can remember those data, and they are mostly asked in the MCQ exam. So just remember, turnover time is a time taken from time taken by a cell to start the division and pass through the entire epithelium. And there are different times, there are different turnover times at the different places of the oral cavity and also in the body, okay, in the skin, in the gut, okay. Okay, after that, in this stratum spinosum, they are the prickle cell layer, okay. So they are a prickle cell layer. Prickle means like spine. And acanthe, okay, so acanthe, they are prickle like, okay. So this is derived from the acanthe, derived from the Greek word, okay. Acanthe means prickle like, prickle cell layer. After that, spinous cell that makes the layer irregularly polyhedral and larger than the basal cell, okay. So the spinous cells are polyhedral and they are larger than that of the basal cell. After that, shrinkage of the cell during this tissue preparation, causing them to separate but remain in the contact only at the desmosomal point, okay. So this is bit similar with that of stellate reticulum, okay. So in the stellate reticulum, there I have told, okay. So with the presence of the glycosam and glycans in the enamel organs, okay, there stellate reticulum, there is just a glycosam and glycan is present in the intercellular spaces between the cells. So they just takes the water, they just extract the water from the cell, and the cell get shrinked, okay, and they form the star-like structure. So similar pattern is also present here in this stratum spinosum. So there is shrinkage of the cell, okay, during the tissue preparation, causing them to separate but contact at the desmosomal point, okay. Okay, so after that, cochlear or sticker like appearance, okay. So cochlear, okay, so this type of a term they can ask you in the MCQ, cochlear or sticker type, like a structure that has each spine ending at the desmosome, okay. So remember this about the cochlear and sticker appearance. And desmosomal attachment plaques contain the polypeptides desmoplakin and placoglobin. Monoclonal antibodies can be used for the detecting carcinomas against those polypeptides by the immunofluorescent microscopy, okay. So in the desmosomal attachment plaques, we contain different kinds of the polypeptide like desmoplakins and the desmoplacoglobin, okay. So desmoplakin and the placoglobin are the different polypeptides that are present on the desmosomal attachment plaques. And by using the immunofluorescent microscopy, with the help of this monoclonal antibodies, okay, we can just detect the carcinomas, okay, detecting the carcinomas against those polypeptides, okay. So now in this intercellular space, we have a glycoprotein, glycosam and glycans, and the fibronectin, okay. So as the cells are shrinked, okay, so there is a space left between the cells, okay. So that is the intercellular space, and there we can see the glycoprotein, glycosam and glycans, and the fibronectin present in the intercellular space, okay. So you can remember this. This is a MCQ question. After that, the most active layer of the protein synthesis, okay. So this is very important, okay. So stratum spinosum is one of the most active layer of the protein synthesis, and twofold increase in the number of the tonofilament than the basal cell, okay. So in the basal cell, if there is x number of tonofilament, then tonofilament will be 2x in this stratum basal, okay. And with this tonofilament, you just remember one concept, that as you move from the stratum basal to the stratum corneum, okay, so there is increase in the filaments, so the quantity of filament goes on increasing, okay. Okay, so after that, in the terms of number and length, okay, desmosome of the spinous layer occupy more of the membrane of the tongue, gingiva and the palate, okay. Okay, so in those places we have more number and the larger length of the desmosome in the spinous layer, okay. So in tongue, gingiva and the palate, so you can remember those. And all those questions can be asked in the MCQ exam, okay. Okay, so we have completed stratum spinosum 2, okay. And in this stratum spinosum, just you should remember prickle cell layer, shrinkage of the cell connected at the desmosome, desmoplakin, placoglobulins are the polypeptide present at the desmosomal attachment plaques, okay. And they are used for detecting the carcinomas using the monoclonal antibodies and immunofluorescent by the help of immunofluorescent microscopy in the intercellular space. Those are present there, and it is a most active layer of the protein synthesis. Okay, so we have completed this, and after that we will discuss with this stratum granulosum. So now in this stratum granulosum, it is a flatter and the wider cells larger than the spinous cell. Okay, so the cell that was present in spinous cell, okay, that was larger than the basal cell, and the cell that is present in this stratum granulosum is again larger than that of the stratum spinosum. So it is larger than both spinous layer and basal layer. Okay, so cells larger than the spinous layer. Okay, after that this is named for the basophilic keratohyalin granules. So basophilic keratohyalin granules, this is a very important point and structure found in this stratum granulosum. Okay, so basophilic keratohyalin granules, okay, they are present in this stratum granulosum, so they are named so granulosum. Okay, and they are blue staining from the HNE. Okay, so they are blue stained. After that, nucleus shows the signs of degeneration and the pycnosis. So nucleus shows the signs of degeneration and the pycnosis in this stratum granulosum layer. After that, synthesis of the protein is decreased here, okay, in stratum granulosum, but it is more in the stratum spinosum layer. Okay, so that is a distinguishing feature of the stratum spinosum. After that, the next one, tonofilaments are dense in quantity and seen in the association with the keratohyalin granules. Okay, so as I have told earlier also, so tonofilament will increase, okay, as you move to the upper layers of the cells. Okay, and they are seen in association with the keratohyalin granules. Okay, so keratohyalin granules means basophilic keratohyalin granules that are present in this stratum granulosum. So after that, membrane coating granules fuses with the superficial membrane and discharge it continues to the intercellular membrane. Okay, so those granules, so those basophilic keratohyalin granules that are present, okay, so the membranes of those granules, they just get fused. Okay, so they just get fused with the superficial membrane, and those content that are present in those granules are discharged into the intercellular space. Okay, so this is a pattern how there takes place a secretion of the content of this granule. After that, this most important, okay, this is a very important of this stratum granulosum layer. Okay, and also in some book you can see this outline, this granule, okay, this outline bodies, okay, that I am going to discuss, also present in this stratum spinosum layer, but I have clear a concept here. Okay, so you just listen to me, and this is very important. So there is a lamellar granules. Okay, so lamellar granules, a small organelle. Okay, it is a kind of a smaller organelle, also known as keratinosome or outline bodies or the membrane coating granules. Okay, so they are the names of this. So there are proper 4 different names of this. Okay, so one is outline bodies, another is a keratinosome, another is a membrane coating granules, and another is a lamellar granule. Okay, so for remaining all those names, I have prepared a mnemonic. Okay, so that is a malkhan. Okay, malkhon you can remember, or malkhon. Okay, so M means, okay, membrane coating granules, okay, and K means keratinosome, O means outline bodies, and L means lamellar granule. Okay, so this is a malkhon. Okay, you can just repeat it and you can remember it. Okay, malkhon, I think it is a surname. Okay, people used to use this surname, so this malkhon. So M means membrane coating granule, L means lamellar granule, K means keratinosome, O means outline bodies, and they formed in the upper spinous and the granular cell layer. Okay, so this is very important. So this is mostly present in the upper spinous layer. Okay, so it is also present in the spinous layer, but in the upper portion, and in the granular cell layer it is mostly present. Okay, so we used to discuss this outline bodies in this stratum granulosum. After that, outline bodies are 250 nanometer in size and contains the glycolipid. Okay, so this is a major constituent of this outline bodies, and those glycolipid, they originate from so those glycolipid are present in these outline bodies, and those outline bodies originate from the Golgi system. Okay, okay, So after that, internal elongated lamellated structure. Okay, so Orlan bodies are internal elongated lamellated structure. Lamellar granules discharge their content into the intercellular space, forming an intercellular lamellar material, that is permeability barrier. Okay, so just there is a advanced language written here. Okay, but I'll just give you a proper overview. Just you think. Okay, so Orlan bodies, or the keratinosome, or the lamellar granule, or the membrane coating granule. Okay, so they are formed from the glycolipid, and they are generated from the Golgi system. Okay, and those glycolipids are secreted on in the intercellular lamellar, in the intercellular space. Okay, and they form the intercellular lamellar material. Okay, so intercellular lamellar material, so this is a one technical term. Okay, you can use during answering the question. Okay, so this lamellar bodies, okay, they just secrete the glycolipid in the intercellular space, and they form the intercellular lamellar material. Okay, and they intercellular lamellar material, okay, those secretion are of glycolipid. So the glycolipid uh, are responsible for forming the barrier. Okay, and that is a permeability barrier. Okay, so those glycolipid are the secretion of those lamellar bodies, so they form the permeability barrier. And after this permeability barrier is formed, okay, so there is thickening and cross linking present between the proteins, and calcium deposition and the cell death take place, and thickened keratin layer is formed. Okay, so how there is a formation of the keratin layer, stratum corneum layer in the upper portion of this granulosome is that. Okay, so this Orlan body secretes the glycolipid that forms the permeability barrier, over which there is a cross linking and thickening. Okay, over which there is a thickening and the cross linking of the proteins, and upon which those proteins there is a calcium deposition. Okay, and with the deposition of the calcium there takes place the cell death, and after cell death there is also deposition of the keratin, and it forms the thick keratin layer. Okay, and that comes in the layer of the stratum corneum. Okay, so that is a physiology of this Orlan bodies. Okay, you can remember this. Okay, after that, this Orlan bodies, we have the main other granule that is present in this stratum granulosome. Okay, and this is important granule, basophilic keratohyaline granule. Okay, so that is the most important granule present in this granulosome. So we will discuss with that granule. So keratohyaline granule, okay, so it is 0.5 to 1.5 micrometer, but this Orlan bodies were 250 nanometer in size. Okay, so these data are important, you can remember those data. So 0.5 to 1.5 micrometer in dimension. After that, in light microscopy, we used to tell basophilic granule, and in the electron microscopy, we used to tell electron drains. Okay, so they are MCQ questions. They can ask you in the MCQ questions. Okay, after that, they are irregular in shape and synthesized by the ribosome. Okay, so their granules mostly contain proteins, and the proteins are secreted by the ribosomes. Okay, after that, we have two different types of the keratohyaline granule. One is irregular and the angular. Another is a regular type. And irregular type, they are associated with the tonofilament, but this regular type are not associated with the tonofilaments, and they are absent in non-keratinized mucosa, and they are surrounded by the ribosome. Okay, so just you should remember. Okay, we have keratohyaline granules. Okay, and also one point is present here, they contains the filaggrin protein that forms its bulk, and laurycin sulfur rich in protein. Okay, so this filaggrin protein and also this laurycin sulfur that is rich in protein, okay, is also present in this keratohyaline granules, and it is also present in corneum layer. Okay, and I will be discussing with those protein here in stratum corneum, as I have written here about the filaggrin. Okay, so just wait for a while. So in keratohyaline granule, just remember. Okay, so they are the granules, basophilic granule. Okay, and electron lens in electron microscope, that is a terminology. And irregular shape, we have two different types, irregular and regular. Irregular is associated with the tonofilament, and regular is not associated with the tonofilament, and it contains the filaggrin protein and laurycin sulfur that is rich in the protein. Okay, so they are the MCQ question. Okay, so they can ask you in the MCQ. After that, in the most superficial part of the granular layer develops a cornified cell envelope on the inner or intracellular aspect of their membrane, and they are called cornified cell envelope. Inner unit of the cell membrane thickens. Okay, so there is a formation of the cornified cell envelope in this uppermost layer, superficial part of the granular layer, and this cornified cell envelope means inner unit. Okay, so the cell membrane's inner unit is thickened. Okay, and they form the envelope like structure, and the inner unit of the cell membrane it get thickened. So inner or intracellular aspect of the membrane it becomes cornified, okay, and thickened. So they are called cornified cell envelope, and this is MCQ question. After that, important protein contributing to this structure, cornified cell envelope, is the involucrin. Okay, and involucrin and loricin, okay, become cross-linked by the enzyme transglutaminase. Okay, forming the 10 nanometer high resistant electron dense cornified envelope just beneath the plasma membrane. Okay, so the function of this cornified cell envelope is the resisting the it is just for protection, okay, from different kind of forces. Okay, so it act as a resistance. So in this cornified cell envelope, as I have told, there is a thickening of the inner unit of the cell membrane on the superficial part of the granular layer. Okay, thus this much you should remember: on the superficial part of the granular layer there is a thickening of the inner unit of the cell membrane, and that is called cornified cell envelope. And the protein that are mostly contributing in this formation is the involucrin, also loricrin. Okay, so this involucrin and loricrin are cross-linked by the enzyme transglutaminase, forming the 10 nanometer forming 10 nanometer high resistant electron dense cornified envelope. Okay, so these are quite vast, okay, terminology, and you shouldn't have to be bothered in those terminology. If you get it, then it is good. If you not get it, just remember just 2-3 point. Okay, so cornified cell envelope, involucrin, loricin, transglutaminase enzyme. Okay, so they can ask you in the MCQ. Okay, and they won't ask you this in a university level examination. They are for the MCQ exams. Okay, so you can just remember those if you want to remember it properly. Then just remember those words. Okay, after that, in non-keratinizing oral epithelium, small organelle similar to the lamellar granule forms. It is a circular and amorphous but elongated and lamellar in a keratinized. Okay, so this in non-keratinized oral epithelium there is also a small organelle like that of the Orlan bodies is present in the non-keratinized oral epithelium too, but it is circular and amorphous. Okay, this is present in the non-keratinized but elongated and lamellar in the keratinized. Okay, so we have completed this stratum granulosum, and I think we have been confused a lot in this chapter, but what to do in this chapter? Okay, so this is a quite big chapter, and also we should have to study this oral mucosa in detail. Okay, this is very important chapter, so we can't miss the every point. Okay, and those points are written in our bands. Okay, so I can't miss those points. Okay, so I have written all those points properly in my notes. So after listening to those lectures and studying those notes for the several time, you will be master in this oral mucosa. Okay, so don't get much worried in this chapter. And this stratum corneum, it is very easy to remember. So stratum corneum, it is also called keratinized squamate, okay, which are larger and flatter than granular cell. Okay, so the cells in this stratum corneum are more larger and more flatter. Okay, in this stratum corneum, and the thickness varies in different sides of the oral cavity. Okay, we can remember this. After that, acidophilic cornified layer composed of the densely packed filaments developed from the tonofilaments coated by the basic proteins of the keratohyalin granules, filaggrin, derived from the keratohyalin granules. Okay, so just remember from this point that stratum corneum they are acidophilic, but in granulosum they were basophilic due to presence of the granules, and those acidophilic layer. Okay, and this stratum corneum, as it is acidophilic, also in this layer we have densely packed filament present there. Okay, we have densely packed filament. Okay, so we have filament, so densely packed filament is present there, that is derived from the tonofilament, and those filaments are coated with the basic proteins, and those basic proteins are the filaggrin proteins, and those filaggrin proteins are derived from the keratohyalin granules. Okay, So I think you have been confused a lot in this line, but once listen to my word again. Okay, so what I am telling that this stratum corneum is acidophilic, okay, and it contains the densely packed filaments, and those filaments are coated with the basic proteins, and those basic proteins are derived from the keratohyalin granules, and those basic proteins are filaggrin. Okay, so this is very important. After that, cells are densely packed with the filaments. Okay, as I have told previously, with the uh, when you go up outward from this stratum basal to the stratum corneum, so filament will increase in number. Keratinized cells become compact and dehydrated and covers the greater surface area than does the basal cell. Okay, so this keratinized cells, okay, so as I have written here, calcium deposition and the cell that take place there. After that, there is a keratin layer deposition. Okay, so keratin deposition is there. So similar thing is written here, keratinized cells become more compact and dehydrated. Okay, so they become dead and covers the greater surface area. Okay, greater surface area than does the basal cell. So this line is written in Orbans. After that, it doesn't synthesize the protein. Why to secrete protein in the outermost layer of the epithelium? Okay, so you can just clear your concept like that way. Okay, so there is no use of the protein in the outermost layer. After that, it is thicker and denser plasma membrane is present in this stratum corneum. Okay, so this thicker and denser plasma membrane, you can just remember like this way. So cornified cell envelope is present in the superficial layer of the granular layer. So that is a form due to the thickening of the inner unit of the cell membrane. Okay, so there is a thicker or the denser plasma membrane in this stratum corneum. Okay, so we have completed for today. Okay, I think today's video was quite vast. Okay, and I have tried to discuss with all those points properly so that you can catch every point in easy way. But what to do? Okay, I can just try, and but you should work hard to understand all those points. Okay, take the proper overview of this chapter. So if you are confused again, so watch this video for more than one, two times. Okay, yeah, more than one time. So how you are comfortable, you can just think uh, and you can do that. Okay, okay, so in the next video of this oral mucosa, we will be discussing with the non-keratinized epithelium and other remaining topics. Okay, so we have a lot of topics remaining in this oral mucosa, and I will be covering all those topics in upcoming videos. Okay, so you shouldn't have to be worried in this chapter, and you should prepare well with me. Watch those videos and read those notes for us several times. Okay, so we'll meet in the next part of this oral mucosa. Till then, stay happy, stay healthy. Tata, bye bye, thank you.